Hello learners, this is Mrs. Garvita Joshi, Associate Professor, Mahakal Institute of Pharmaceutical Studies, Ujjain. Today we are going to learn about receptors. After this session, you will be able to know the different types of receptors which are present in the body, understand how these are classified, and explain how these receptors function. First of all, coming to the definition, how the receptors are defined. Receptors are defined as the specialized areas of cell to which a drug binds. In biochemistry and pharmacology, these are the chemical structures which are generally composed of protein that receive and transduce the chemical signals that can be integrated into the biological system. When such chemical signal binds to a receptor, they causes some form of either cellular or tissue response. When the drug binds with the receptor, these interactions are known as drug-receptor interaction, and the effect of drug is attributed or depends upon two factors: one, affinity, and second, intrinsic activity. Affinity is the ability of a drug to bind with the receptor to form a complex which is known as a drug-receptor complex, and intrinsic activity depends upon formation of this drug-receptor complex. It is defined as the ability to trigger a pharmacological response via the drug-receptor complex. Depending upon this, the drug can be a full agonist, full antagonist, or partial agonist. Full agonist is one which binds with the receptor and shows a maximal response in positive direction. Full antagonist, it also binds with the receptor but shows a maximal response in opposite direction to that of agonist. Partial agonist, which is having a sub-partial binding with the receptor and hence it shows a sub-maximal response. Isolation and identification of receptors. The membrane receptors can be isolated from the cell membranes by complex extraction procedures which involve the use of solvent, detergents, or they can be isolated by affinity purification. These receptors which are isolated, their structures and actions can be studied using different methods like X-ray crystallography, NMR, circular dichroism, and real polarization interferometry. Along with this, the computer simulations of the receptors have been used nowadays to understand about their mechanism of action. Coming to the classification, the receptors are classified based upon their position, and depending upon this, there are of two types. One, which are known as transmembrane receptor, cell surface receptor, or cell-specific receptors, which are generally located on the surface of the cells. Second, intracellular receptors, which are generally located inside the cell. Coming to the first receptor, that is transmembrane or cell surface receptors, they perform the signal transduction, convert an extracellular signal into an intracellular signal. There are three main components of this transmembrane receptor: external binding domain, a hydrophobic membrane spanning region, and intracellular domain which is located inside the cell. This is the extracellular domain, this is the intracellular, and this is the one hydrophobic domain which crosses the cell. A portion lies outside and a portion lies inside. The intracellular receptors, as the name indicates, located within the cells, also known as cytoplasmic or nuclear receptors, which are found generally in cytoplasm or in the nucleus. They generally respond to the hydrophobic ligand molecule as it can easily diffuse inside the cell and react with the intracellular receptors in the cell. Example of this type is a steroidal hormone. Now, coming to the first type that we have seen, that is the transmembrane of cell surface receptors. These cell surface receptors are divided into three subcategories: ligand-gated ion channel, G-protein coupled receptor, or enzyme linked receptors. Take a pause and think why the ligand-gated ion channels are named as such. Write on a piece of paper, memorize in the mind, and let's see about them. The ligand-gated ion channels are known as such because they generally contains a channel through which a specific ion flows. So, these are the protein molecules which form water-filled pores which spans the membrane. A receptor binds with the ligand, opens a channel which allow a specific type of ion to flow. And this ion is generally sodium, calcium, chlorine, magnesium, etc. This is the diagrammatic representation of how an ion channel opens when a messenger binds with it. This is the closed channel. When messenger binds with the receptor, it opens a channel which causes a particular ion to move through this, resulting in change in the electrical properties of the cell. The effects which are observed are there can be increase in sodium and calcium or decrease in sodium and calcium levels. Increase results generally in excitation and decrease results in inhibition. Another, there can be increase or decrease in potassium level, again resulting in inhibition or the excitatory effect of the cell. And third main ion which flows through them is chloride. Again, chloride channels can be open or they can be closed, resulting in inhibitory or excitatory effect of them respectively. So, we can see that the concentration changes of ion can result either in excitation or inhibitory effect of the cell depending upon the type of ion which flows through. The second subcategory is known as G protein coupled receptor. The drug binds with a protein which is known as G protein, that is guanyl protein, activates or interact with them and activates either an enzyme or channel which will further activate sequence of events. These are further subtypes of G protein which acts through adenyl cyclase pathway, either activates or inhibits it. Second one is the phospholipase CYP3-DAG pathway, and third one is opening of channel, which is particularly calcium and potassium channel. These are the examples of the subtypes of GS receptors. GS examples are beta receptor, GI muscarinic receptor, GQ alpha 1 H1 M1 receptor, and GO the channels in heart. This is a diagrammatic representation of how the beta receptors is activated by a norepinephrine, resulting in activation of adenyl cyclase, which further activates inactive protein kinase to active protein kinase, resulting in opening of calcium channel, which further releases calcium ions. This calcium ion activates troponin, resulting in enhanced actin-myosin interaction and further increased cardiac contractility, force and velocity, which are the final results of binding of norepinephrine to beta-1 receptor. The third subtype of transmembrane receptor are enzyme-linked receptor, which are also known as catalytic receptor, and these are the cell surface receptors with intracellular domains that are generally associated with the enzyme. That's why they are known as enzyme-linked receptor. When a ligand binds to the extracellular domain, a signal is transferred through the membrane and activates enzyme which leads to response. This is an example how a tyrosine kinase enzyme is activated by binding of a drug, resulting in conversion of inactive to active form and further activation of different proteins into the cell. Coming to the second subtype, which is intracellular receptor, as we have discussed it, these are the receptors which are present in the cell, also known as nuclear receptors, can be present either in the cytoplasm or they can also be present in the nucleus. This transduce the signal by modifying the gene transcription and have an important role in endocrine signal and metabolism regulation. They can directly bind with the DNA and regulate the gene expression. Certain examples are steroid hormones and glucocorticoids. The pathway is shown how they functions. The steroid hormone crosses the cell membrane, reacts with the receptor which is located in the cytoplasm, forms a receptor hormone complex. This complex then crosses the nuclear membrane, reaches nucleus, activates DNA, mRNA and result in the synthesis of proteins. So this is the pathway how a cell receptor or intracellular receptor functions. These are certain examples of different drugs which binds with different type of receptors. Let's have a look. First one, you will see here the non-steroidal anti-inflammatory drugs, angiotensin converting enzyme inhibitors, they binds with enzymes. Beta blockers, adrenaline, it binds with GPCR. Ketamine binds with ligand-gated ion channels. Insulin binds with enzyme-linked receptors. Calcium antagonist generally binds with voltage-gated ion channels. Antiviral drug, the DNA can be a target, and glucocorticoids, they interact with the nuclear receptor. So these are the different drug targets. They can be enzyme, they can be channels, they can be GPCR, and they can be intracellular receptors. So this is how the drug binds with the receptor and elicit a pharmacological response. Coming to the summary, we have seen 4 types of receptors, that is ligand-gated ion channels, also known as inotropic receptor, G-protein coupled receptors, that are metabotropic receptors, kinase-linked receptors or enzyme receptors, and nuclear receptors, which are generally located in the nucleus. This is the time scale, how much time it is needed to elicit the response. Ligand-gated ion channel, within millisecond response is produced, GPCR in seconds. Kinase-linked receptor, it takes hours to produce the response, and nuclear receptors again hours. And certain examples, nicotinic in ion channel, G-protein coupled receptor, muscarinic receptor, kinase-linked receptor, cytokine receptors, and nuclear receptors, they are estrogen receptors. So these are certain examples. So that's all about the receptors. Hope you have a happy learning session and happy learning further. Thank you.